Welcome back. Now we're going to talk about Unit 4, which is really all about the cell. So, a long time ago, we didn't know why we got sick or what we were made of. And then some wonderful people made some great observations once we started developing microscopes, and they found what is called the cell. With that came unified cell theory. So, what this says is that all living things are composed of one or more cells. And cell is really the basic unit of life. It is the smallest unit of life. Um, all known things are made out of cells. All new cells are made by pre-existing cells. And then lastly, the cell is the most basic unit of structure. Some important things to know. We have what's called prokaryotic cells. These are small, simple, and normally single-cell organisms, such as bacteria. They do not contain a nucleus or any other organelle other than a ribosome. So once again, that's a prokaryotic cell. I like to remember prokaryotic as pro, as meaning before, and before there was a lot more stuff we need to learn about, which we talk about next in the eukaryotic cells. So eukaryotes, they're larger, they are more often multicellular, so larger organisms, um, and really they have a lot more structures. So, real quick, the common cell structures between these two different types of cells would be the cell membrane. The membrane is really most important. I kind of mentioned it in the last summary. It's the phospholipid bilayer. It's semi-permeable. We'll kind of get into that in a little bit later. It also has cytoplasm. This is like the intracellular space. It's the fluid within the cell. There's a lot of things contained within it, but the cytoplasm is really where all the chemical reactions occur within the cell. Also, we have DNA. This is the instructions for protein synthesis. It also has the hereditary material that the cell needs in order to reproduce. We also have proteins, so raw materials that are used to build structures, and then last but not least, ribosomes, and these are the protein builders. Okay, so eukaryote cells, they have organelles. These are all membrane-bound, tiny little functional systems within the cell. So first, we have the nucleus. It is right here. In it, it has DNA. It could easily be assimilated to like a control center in a building. It is surrounded, you can't really tell here, but it's surrounded actually by a double layered membrane called the nuclear envelope. Inside it, there is the nucleolus right here. And this is actually not membrane bound, but it's actually an aggregate of molecules where ribosomes are assembled. And like we mentioned, ribosomes are really the protein little producers, so those are important. Next we have mitochondria. That's not it. Mitochondria, I'm reading it backwards, right here. Okay, so mitochondria, this is commonly referred to as like the powerhouse of the cell. These really take in sugar, so carbohydrates, and they make it into usable ATP. In plant cells, there's also chloroplasts. These take in energy from the sun and convert it into usable ATP, but this is a plant cell, so you don't see any of those here. Next, we have the endoplasmic reticulum. We have that right here. I don't know, can you see that? Endoplasmic reticulum. This does a lot of different things. It assembles proteins and other cellular components. So rough ER, this actually has little ribosomes on it. That is contributing to making proteins. And then smooth ER typically tends to make lipids, but also it can help to detoxify things like alcohol, drugs, anything in your system that needs detoxifying. Next, we have the Golgi apparatus, which is right up here. Looks like that. It's the packaging and shipping center of the cell. So what it will do is any proteins that are going to be moving outside of the cell, it'll take those in once they're created in the rough ER, and package them kind of for transport. It's like UPS almost. Um, so what it packages them is vesicles or vacuoles. So vacuoles are primarily for storage. Vesicles would be for, you know, moving outside or transport. These can fuse with the plasma membrane or other organelles like the endoplasmic reticulum or the Golgi, allowing them to then be transported there. Okay, next we have lysosomes. I hope they're on here. Here we go. Right up here. These are only found in animal cells, and what they do, they're specialized vesicles that actually have digestive enzymes inside. They actually actively pump in hydrogen ions, lowering the pH in order to essentially eat and dissolve maybe waste materials or bacteria, anything that the cell doesn't want inside it necessarily. Destroys internal invaders. I thought that was kind of cool. Okay, next we have cytoskeleton. So, we don't have anything necessarily specific cytoskeleton, but these microtubules, that would be part of it. Essentially, they contribute to the structure of the cell. They help it maintain its shape and then also function to help transport things around the cell. If you think about it, the cell without these might be kind of a liken to floating around in outer space without anything to push off of, whereas these kind of give it its structure, allow things to be transported in certain ways, etc. Okay. Next, we have cell membrane. This is important. It's the thing that encapsulates the entire cell. So it's primarily consisted of the phospholipid bilayer. So the hydrophobic tails are going to be on the inside, and the hydrophilic heads are going to be on the outside. So what does that look like? Just in rough terms, let me draw you what it would look like. Okay, so hydrophobic tails aligning on the inside, hydrophilic heads. This would then be like the inside of the cell, and this would then be oriented to the outside of the cell. So you can see how this would maybe be permeable. These are not in a fixed place, but they're constantly moving. In fact, in this cell membrane, sometimes there's even cholesterol, which would be unsaturated fats. So they're kinked, they allow for movement and for these phospholipids to not stack in the event of getting colder. So animals like polar bears and stuff would obviously have a lot of these to really prevent cells from freezing up and dying in the extreme temperature. So good to know, good to know. They're semi-permeable, I kind of mentioned that. So small gases and hydrophobic molecules can pass through easily. In the event that you're not a small gas or a hydrophobic molecule, you would then have to either be actively transported with a transport protein, which are also found in the cell membrane, or channels have to specially open for you, or potentially you need to be engulfed by the cell through not endocytosis, or expelled through exocytosis. We'll talk about that a little bit more here as we go on. Okay. Kind of talked about that. Other things that are found in the cell membrane, there's actually a lot. There's carbohydrates, proteins, sterol, so cholesterol, like I mentioned. So proteins, they assist with transport. Carbohydrates, they actually give cells their identity. So this is actually the part that identifies different human blood types, for example. So that's important to know. Proteins, they aid in communication, so they can receive signals from external. So external sources will send communications, proteins will receive them. These will then in turn be interpreted and dispersed within the cell. Proteins also can assist in metabolism, so they can form enzymes. These allow the cell to grow, obtain energy easily, or eliminate waste. Okay, next we're going to talk about diffusion. If you have ever dropped just a droplet of food coloring into a glass of water, you've kind of watched it. I mean, you don't even have to stir it. If you watch it, it'll swirl about and eventually it'll disperse to the point where there's an even color in the entire glass. That is actually a perfect example of diffusion. So what this is these solutes that are present in the solvent or the solution exist in a high concentration in one spot. However, they're all moving due to kinetic energy, and in time, they're going to all spread apart to be more or less evenly distributed in the entire solution. This is called Brownian motion. So that is simple diffusion. Likewise, things are going to move across their gradient. So if you have a lot of something here and nothing over here, all of these are more likely to diffuse towards the area you do not have anything in. And almost like a tidal wave type action. I don't know if you've ever been to the wave pool at Wild Waves, but same idea. The water is going to rush where there's less water. Same thing with diffusion. That's the same idea. So molecules use diffusion to get into or out of cells as well. So passive diffusion would just be just that. I mean, no energy on the cell's part, no action. It just is get in or get out accordingly, according to the concentration on either side. Next, we have facilitated diffusion. So I kind of talked about it, but pores in the cell will actually open up to allow the larger molecules to then diffuse in or diffuse out according to its concentration gradient. This would be facilitated diffusion. So here, even though a pore is open, there's actually no real energy expelled by the cell. These things are able to come in on their own. Next, we have active transport. So what this looks like is a perfect example, lysosomes. So lysosomes actually maintain a very low pH internally, and how they do that is they import by actively transporting hydrogen ions into the inside of this organelle. That is not active diffusion or simple diffusion. If it was, it would be equal as inside as outside of the lysosome. So these require transport proteins. It also requires energy. One example of this would be endocytosis. That's when a cell takes like this engulfing like arm and just grabs whatever it is it needs to grab and then brings it into itself, engulfing it in this pocket of a membrane. So then you have like a vascule floating around inside the cell of whatever it was the cell engulfed. Likewise, you have exocytosis, so that would then be that bubble hitting the cell wall on the inside of the cell and then opening up and spilling out its contents outside of the cell. Ooh, phagocytosis and pinocytosis. I did not talk to you about those. So phagocytosis, that is when endocytosis is occurring, but it's taking in larger molecules. So maybe like a whole other molecule outside the cell, maybe a bacterium, whatever. Then we have pinocytosis. This is normally fluid, small molecules. Really, how I remember pino would be like a pinprick, and then phagocytosis sounds like, I don't know, it just sounds like eating a lot more, so that's how I remember it. Okay, Next one, we have osmosis. So this is the tendency of water to move across the cell membrane in a way that establishes equal concentration of solutes. So sometimes, due to that semi-permeable cell membrane, sometimes you have a higher concentration of whatever it is, solutes, inside the cell or outside the cell. So in an effort to maintain equilibrium, the cell will free the water or take up more water to kind of evenly distribute that. This is typically not very good for the cell. So we've got three different types of stages here. The first one is hypertonic, right here. So as you can see, there's a lot more, in this case I drew sodium ions. There's a lot more sodium ions outside the cell as there are inside, so the water will come rushing out in an effort to equalize this gradient here. We have isotonic where it's equal. This is most ideal for animal cells because the water can go in and out freely and it's equal. The cell is happy, it's maintaining homeostasis. And then we have hypotonic. So there's a higher concentration gradient inside the cell. So what will happen is water will come rushing in and unfortunately cells aren't made to handle that and they will typically burst and die. Not a good way to go. Distilled water is the ultimate hypotonic solution because all of our living cells, they contain solutes, be it organelles, be it ions, whatever. So having that introduced into your body is definitely pretty lethal. Likewise, brines and syrup are hypertonic, so they've got a lot more solutes present. And then saline is isotonic. One quick fun little fact, plant cells are protected in hypotonic situations due to the cell wall. So that is a key differentiating structure between animal and plant cells. So the cell wall is more like a rigid structure than the cell membrane. It's literally like a wall. So because of this, it forces water back out of the cell once it is completely turgid. In hypertonic situations, however, the membrane is torn away from the cell wall and this results in the cell death. So it tends to be vice versa for animal and plant cells. All right, so that is my conclusion to Unit 4 summary. Thank you for watching and good luck on the exam.